We're going to move directly to the environmental issues with Andreas Kurz. I'm going to ask Andreas to come here. So Andreas is the head, you stay here, right? So yeah, just for organization, we invite all the speakers after they finish their speech to come here to have questions and remain on the panel. So Andrea is head of the Laboratory for Stem Cell Research and Knowledge Management at the Berlin Brandenburg Center for Regenerative Therapies at Charity Hospital. Since 2007, he's a coordinator at the European Human Pluripotent Stem Cell Registry and member of the European Group on Ethics since 2017. He accumulated various academic professor roles from Seoul in Korea to Berlin in Germany, passing by Georgetown in Washington, D.C. And finally, he has been the CEO of various stem cell and genetic diagnosis companies. So you have the floor, Andreas.
Thank you very much. I would like to right in the beginning, thank the organizers and especially Francois Hirsch for inviting me, making this possible. And he also asked me to talk about environmental issues, which is not my specialty, but I'm trying to put on some of these issues which I think might be important and worthwhile to be discussed. So this graph I took from a publication from the high-level group of scientific advisors of the European Commission. And it shows that the different degrees in which we influence environment by using natural mutations or artificial mutagenesis to develop plants or animals for our benefit. So it started many thousand years ago. Lamarck was already mentioned, Mendel. Next step was mutational breeding by, for example, radiation or chemicals, marker-assisted selection, and transgenesis. And now we have genome editing. The main difference with the previous technologies is that we can very specifically modify a genome and reduce the need for selection after that. There are, of course, other differences between what you can do with genome editing and what you could do before. And to find that out, what is actually different now with genome editing is a task we all need to do. It's if you think just creating mutations, that's not so different from what we have been doing before. But what we can do, and I will focus on that, is providing methods for gene drive. So we can basically sustain a mutation in a population which then becomes dominant. And we can also use these technologies to create artificial organisms or new species even. And I will try to talk about this a little bit. And with that, there are, of course, a lot of promises connected. And some are listed here. This, again, I took from this source, Mathematical Ecology Research Group. There are promises in agriculture, of course, by, for example, pest management or using non-toxic herbicides and pesticides by creating plants which are sustainable, resistant, and very proliferative. We can apply this in human health, for example, by creating immunizing animal reservoirs of disease, or we can control vector-borne diseases, which has already been pursued. We can, of course, aid threatened species in their survival fight. We can also control invasive species using these gene drive technologies. And there are, of course, with all these promises, there are also certain new challenges or risks. These are completely new tools for ecology, which we haven't used before. Yeah, and we need to find out what is the consequence of doing that. And one issue is how safe safely can we control these gene drivers and transgenesis created by gene drives. And I'll list here a few of these challenges. So with gene drives and the development of new organisms, you have a high depth of intervention. So you intervene directly in the genome level, you target that technically, and you have a high impact because you target the genome directly, as far as the genome genes determine the phenotype, of course. And then you design an instrument, an organism, which is designed to spread towards irreversible mutagenic chain reactions. So, and the point is here irreversibility and the chain reaction. So it's this wording already implies uncontrollable kind of, at least on the genome level, uncontrollable events. Because of that, the level of not knowing what will happen is just technically magnified. But there are also non-technical issues which are magnified on the ecological level. There's a high potential for exposition, especially when the gene drives are released in the wild, in nature, and because they are mobile. You cannot keep them encaged in a certain area, and they self-reproduce, they're persistent, self-sustaining, enhancing the contamination of unclear fate, and they're usually not self-limiting, although one can, of course, think about technologies where even these gene drive technologies might be self-limiting. It's difficult or it's challenging to monitor these new plants or animals, and it's questionable whether it's reversible. There's a high depth intervention, not only technically, I mentioned that already, also on the ecological level, on the ecosystem. Another issue about gene editing has already been mentioned several times. It's cheap, it's easy to do. You can buy a kit to do it in your garage. And you can also use it, because of that, to change simultaneously several genes at the same time. Something which occurs also naturally through evolution, of course, but here you can do it very quickly. And evolution is usually not so quick. So you introduce something in an environment which has not been adapted to an evolutionary event by changing a lot of genes at the same time, creating basically a new species if you do that in many, many genes. But you also have the potency to eradicate species, at least regionally, either because you want to eradicate them or you delete or deplete species from prey. So they will die out. The stability and safety, and now I come to other issues of the technology, are questionable. There is not enough knowledge about technical failure. There's not enough knowledge about side effects. And the continuation of evolution, which I mentioned, on the genetic level is possible. And also there's a possibility, which we don't understand completely, of cross-species transfer, at least in microorganisms, that has been shown and was already mentioned. So this is a lot of there are a lot of promises, of course, but there are, as I mentioned, a lot of issues to consider. And there are some questions I would like to bring up. And the first one is, should we just do a basic risk-benefit analysis and then go ahead if the balance is shifted towards the benefit? Or should we adhere to the precautionary principle that has already been mentioned? And the precautionary principle, it seems, needs to be applied, but that also needs action. And what actions do we need to do to apply this precautionary principle, which is useless if it's just saying, well, we shouldn't do something, but because there is some risk. But what can we do to reduce the risk, to answer the question whether we should do it or not? A driving factor for the application of gene editing in this environmental area is commoditization. It's a general issue in biology, but here specifically, these things make a profit, and this is a driving force for their application. And the question is how and whether we can control it. Do we want to control it? Can it be controlled at all? That this commodification drives this issue and not other issues, scientific or ethical issues. Another issue which I just want to mention without going into deeper is that we are working with CRISPR-Cas technology in the lab as well in human cells. The RNA, the DNA we are targeting is human DNA. So we have floating around these protein molecules of CRISPR-Cas and the guide RNAs and the target RNAs, which are inhaled, of course, by people who work with that. And the issue is whether we don't know, is there a health issue when you inhale these molecules? Do they enter cells or not? Can that cause some somatic problems? So the observatory which was recently published in this paper, right? A global observatory for gene editing. I like this picture they have there. Is something which reminds me of our group here. What they propose is to implement three structural, let's say, issues. One is to create a clearinghouse, a collection of statements and regulations which are out there to track conceptual developments, also scientific developments in the area, and be a vehicle for meetings and outreach. Just what I think we also should do. Already mentioned was we have working groups which are to be implemented, and codes of conduct or guides, governance guides, which are kind of these soft regulations, which have been already talked about. As you said in the beginning when you introduced me, I'm running this registry for human embryonic stem cells, and 20 years ago, these human embryonic stem cells were a hot issue in science. Also, actually. Not only among scientists, but also publicly debated quite hotly. And the European Commission thought, well, we can create transparency in the field by making this registry, by register all these human embryonic stem cells and make these available to everybody to look at how many are there. Is there a slippery slope where more and more embryonic stem cells will be derived or not? One issue what one could think about is also as part of this group is to create a registry of these genome editing experiments which are being done, research being done, either in the human field or also in the environmental area. So thank you very much.